So I guess I'd call myself a statistical geneticist by training. So I'm not an epidemiologist by training. I only really got into epidemiology when I moved to the University of Bristol in 2007. So I wanted to kind of give a talk on what the geneticists ever did for us in epidemiology. So I want to start the story back in the 1860s in a small vegetable garden in a monastery in the Czech town of Brno. And many of you will know that that's where the famous Augustinian monk Gregor Mendel conducted his experiments that elucidated the laws of heredity. And so he, in particular, did a number of experiments over several years on the humble pea plant. And he looked at 7 different traits that you see on the left-hand side of the screen. And it was rather fortuitous because the pea plant is a great model for understanding Mendelian genetics. So you've got a number of very clearly distinct binary traits, which happen to follow what we call now Mendelian principles. And the genes underlying these traits, as it turns out, happen to be on different chromosomes or very far apart on the same chromosome, which is really useful for his second law that we'll talk about in a second. You can also manually cross-pollinate and self-pollinate the plants. They have very short generation times and they have large numbers of offspring. So a really great kind of model for demonstrating genetics. And so he basically came up with 2 laws of inheritance. We refer to them as the law of segregation and the law of independent assortment, although he obviously didn't at the time. And so the law of segregation is that alleles, so an allele is a different form of the same gene. Sorry if I go into genetic speak from time to time. I'll try not to do that too much. But the law of segregation states that alleles separate at meiosis, and then a randomly selected allele is transmitted to the offspring. Law number 2, the law of independent assortment, is that alleles for separate traits are transmitted independently of one another. So that's basically what's happening at one position in the genome is independent of what happens the other position of the genome. I wanted to put these up now because we'll come back to them a little bit later in the talk. So he published the results of his experiments back in 1865 in a really obscure journal, and they were rapidly forgotten. And it took another 35 years before they were rediscovered. Interestingly enough, it's taken until this year to fully work out the molecular basis for Mendel's traits in the pea plants. So the last 3 traits, so I think it's the color of the pods, the shape of the pods, and where the flower sits, these sit in very difficult regions of the pea genome to sequence. And so this paper appeared in 2025 in Nature earlier on this year. So what is that? Like 160 years later, we've finally worked it all out. Okay. So that's the deal with Mendelian traits. So when I say Mendelian traits or Mendelian diseases, I mean where there's a one-to-one relationship between genotype and phenotype. So you can see these things, you can observe these things clearly, right? What about quantitative traits where we've got continuous distribution? Well, this actually elicited a very strong argument at the time. So this is in the early 1900s. So on the left, you have the biometricians, so folks like Carl Pearson, and they believed that the inheritance of quantitative characters was like a blending from you were the result of a blend between mum and dad. And then on the other side, you had the Mendelians, folks like William Bateson, and they're like, Mendelian genetics is for discrete traits, not for continuous traits. And this was a really acrimonious debate that went on for a decade or 2. And then this brilliant fellow, who you might have heard of, Ronald Fisher, came along in 1918, And he published this seminal paper called The Correlation Between Relatives on the Supposition of Mendelian Inheritance. Absolutely seminal paper for genetics and for statistics as well. Do not try and read this paper. It is famously impenetrable. There's a much better summary by a guy called Pat Moran in 1966. Read that one first. Like, I do not understand this paper. It's really obtuse. So Fisher not only made seminal contributions to population genetics, quantitative genetics, he was also a brilliant statistician as well. So you could call him the father of modern day statistics. So if you've ever used an F test, the F named after Fisher, if you're familiar with the concept of variance, he kind of elucidated that in this paper. Even the much maligned p-value, that's one of his as well. Absolutely brilliant guy. He didn't always get it right, though. So famously, he believed that smoking did not cause lung cancer. He was a smoker himself. He actually believed that there was a major gene that controlled your propensity to smoke and to get lung cancer. And so it was only in the 1950s, a guy called Jerome Cornfield published something, his famous Cornfield inequality, that showed that this was just implausible and couldn't happen. So Fisher didn't always get it right, but he got most things right. So in this particular paper, what he showed is that quantitative traits were not the result of just one gene of large effect, but they were due to lots of genes adding together. And so that's what I've tried to illustrate on this slide. So the first panel is when you have one gene, so you have 3 different genotypes, and you might be able to discern them. But as you add more and more genes to the system, you get more and more of these classes. It looks more and more like a normal distribution, then you put a bit of the environment on top of that, and you've got a quantitative trait. So Fisher basically demonstrated, actually, yes, quantitative traits, complex traits, they're subject to Mendelian inheritance as well, just like your pea plant. Okay, so that was back in 1918. One of the big controversies that raged over the next couple of decades was this nature versus nurture controversy. So are individual differences the results of genetics, the environment, or both? And so one of the main tools that geneticists used to work out what was going on was the classical twin design. So here you compare the similarity, phenotypic similarity between MZ twins, so these are monozygotic twins, they're genetically identical. You compare that with the similarity between dizygotic twins who share, on average, half of their genes. And so if the MZ pairs are more phenotypically similar, then that's evidence that the trait is at least partially genetic, and you can work out what the proportions are of genes in the environment. Very common method for estimating heritability in humans. It controls for common environment, but it makes this big assumption called the equal environments assumption. That's basically the degree of environmental similarity that the twins experience. It's the same in MZ and DZ twins. So we can argue about that, we can quibble about that, but that's what it is. And so after several decades of folks doing twin studies, Eric Turkheimer came up with the 3 laws of, well, he called them behavior genetics, but I'm going to call them complex trait genetics because it's basically the same. And this kind of summarizes the situation as we understood it in 2000. So the first law, virtually all complex traits are partially heritable. How do we know that? Well, MZ twins are more phenotypically similar than DZ twins. MZ twins reared apart are more phenotypically similar than DZ twins reared apart. And we can confirm that using molecular approaches as well. So what I'm saying is not just dependent on the results of twin studies, but on a whole heap of other quite clever study designs. The second law, the effect of genes on individual differences is typically larger than the effect of the shared environment. How do we know this? Identical twins reared together are phenotypically similar to identical twins reared apart. And then finally, and perhaps most disturbingly, a lot of variance in complex traits isn't attributable to either genes or the shared environment. How do we know this? Identical twins reared together aren't identical. And this brings to the fore this disturbing phenomenon that's been referred to as the gloomy prospect by Robert Plomin and others. And this is kind of the idea that this component that is not due to genetics or shared environment, it may not just be due to specific factors in the unique environment, but it might just be stochastic, like the slings and arrows of outrageous fortune if you're a Shakespeare fan, okay? And we mightn't be able to properly account for this. So if you're a precision medicine aficionado, the gloomy prospect is saying, you're wasting your time. It's very, very disturbing. George Davey-Smith, who many of you might know, has a great article in IJE on the gloomy prospect. Thoroughly recommend people read it if this is your thing. Here's an empirical study from 2015 by a colleague of mine, Tinka Polderman. And what she did was a massive kind of meta-analysis, systematic review, basically taking every single twin study that she could find for all time. So 17,000 traits, 2,700 publications. And I've ripped this slide from one of her figures. The top panel just looks at the correlation between MZ twins in the hot colors and DZ twins in the cool colors. So you can see that across the range of human phenotypes, the MZ correlation is roughly about twice the DZ correlation, which is consistent with a genetic model. And the MZ correlations, they vary from 0.6, 0.8, something like that. On the bottom panel is the estimated heritability. So heritability is the proportion of variance explained by genetic variation. And so you can see across all these traits, there's substantial heritability right across the board, sort of roughly 50%. In the green is the shared environmental component, much, much smaller. There's some traits there. Social values is the one that kind of sticks up. That looks like, looks very different to the others. These are a little bit more difficult to analyze with the standard twin model because of something called assortative mating that screws up the estimates from twin studies. So that's part of what's going on there. Okay, so that's talking about variance components and heritabilities and things in aggregate. What about the individual genes that are responsible for these individual differences? So these sorts of questions were largely dictated by the technology that was available at the time, okay? And so in the early days, we couldn't do much about it except look for a handful of blood groups and these sorts of things. But with the explosion in molecular genetic technologies, now we could look right across the genome. So one of the two methods that geneticists used to investigate individual genes or genetic variation was called the candidate gene association study. So the idea here is you take your best guess what the gene is going to be involved in a particular trait, and you genotype a genetic marker in that gene, and you see whether it's associated with your trait or your disease of interest. So in this particular example on this slide, they're looking at what's called a single nucleotide polymorphism, or a SNP, which you might have heard of before. So this is just a single change in the genetic sequence. And hopefully you can see from this slide that the A allele is more common in cases than in controls. So we would say that this genetic marker is associated with risk of disease, and the A allele is the predisposing risk allele for that disease. You do the analysis just like in a standard epidemiological case control study. It's not different, okay? It's just that you're using a genetic marker. But that genetic marker, it's just like another variable, okay? So don't ever be afraid of genetic epidemiology or genetic studies. They're really simple. Like, most of the time, you epidemiologists do much more clever and sophisticated things than we geneticists do. It just looks fancy because, I don't know, it's genetics, right? So I want to highlight what was the first robust genetic association with a common complex disease. And the reason why I've chosen this disease is because it'll reappear later, and I work on it a lot. It's quite close to my heart. It's something called ankylosing spondylitis. So it is a common complex disease. So it's multifactorial. There's lots of genes contributing to it. It's a horrible disease. It's basically an autoimmune or inflammatory arthritis that's characterized by a progressive fusion of the vertebrae over the life course. Lots of other nasty symptoms as well. But it also happens to be the very first robust genetic association with a common complex disease. So this was back in 1973. The particular allele was something called HLA-B27. So this is just an immune gene in the genome. The thing that's special about this, though, is it's got a massive odds ratio. So if you have a copy of this thing, you have like 50 to 100 times the risk of having ankylosing spondylitis. And that's why folks discovered it back in 1973 with only 70 patients or something like that. It also means that you can use this in the clinic. So ankylosing spondylitis is very difficult to diagnose early in the disease. It's very kind of non-specific symptoms. And so if you type this thing and your patient doesn't have it, it's very unlikely that they have ankylosing spondylitis. Anyway, we'll talk about that some more in a moment. At the other extreme, the other main gene-finding approach was something called genome-wide linkage analysis. I really like this approach because it's hypothesis-free. Now, I think folks in traditional areas of science get very stressed when things are not hypothesis-directed. I actually see this as a strength. So in genome-wide linkage analysis, what you can do is you can take your samples and with 400 genetic markers, you can look right across their genome. So it uses something called microsatellites and this old technology called gel electrophoresis, which you see on the left-hand side of the screen. Not important. The important bit is you're looking at the whole genome just by looking at 400 markers. And the way it works is you look for what's called co-segregation of a genetic marker in a disease. And so what I mean by that is that the marker and the disease are transmitted together. So if we have a look at this pedigree on the right, so the affected individuals are shaded, and you can kind of see that 4 allele as it gets transmitted down the pedigree, so does the disease as well. Although that fellow right at the end, this granddaughter here who's 1-4, what's going on there? She's got a 4, she's unaffected. Well, she got that 4 allele from her unaffected dad, not her affected mum. So if you look at you imagine this black thing, this black rectangle is the chromosome that's getting transmitted down the pedigree, and you imagine there's a red disease gene and a green genetic marker. You can see that as that chromosome goes down, transmitted down the pedigree, both the disease gene and the marker are being transmitted together. They're co-segregating together. And so we can say, oh, there must be a disease gene somewhere near this genetic marker. So we can look right across the genome in a hypothesis-free fashion, generate something called a LOD score plot. So the y-axis, as usual, is your measure of statistical significance. Your x-axis is the genome. And whenever you get a big peak, you get excited. You think, there's a gene under this peak that has something to do with the disease that I'm interested in. And so this is the first step of something was called positional cloning. So basically, folks would take these pedigrees. Here it might be affected sibling pairs or something. You conduct your hypothesis-free linkage analysis. You get a peak somewhere on the genome, and then you look at what genes are under that peak. You know, we have the Human Genome Project, you know, the sequencing of the human genome, so we know where the genes are. We take a look at what genes are under that peak. We then do a candidate gene association study, maybe some sequencing, and maybe we get lucky and we work out the genetics of that particular trait. Here's an example of, I think, the first disease that was worked out using that approach, something called Huntington's disease, horrible neurological autosomal dominant disease. It was worked out by this positional cloning approach. Here's some others as well. The thing that's linking all these findings is that they're either Mendelian diseases, okay, so remember, one gene, one disease. If you have the risk genotype, you're going to get disease, that sort of thing, or Mendelian forms of disease. So you can see BRCA up there. That's kind of a more Mendelian form of breast cancer. And so when I started out in the field in the late 90s, this was kind of the situation. So this linkage analysis and positional cloning approach had been very successful in terms of working out the genetic basis of Mendelian diseases, but complex diseases and traits, we were not doing well. The reasons for that is that when you have a rare disease allele that has a big effect, like in a Mendelian disease, linkage analysis is great, works super, super well. The problem is that's not what's happening in the majority of quantitative traits and common complex diseases. It's actually common genetic variants of small effect, and linkage analysis doesn't work in that situation. Candidate gene studies also weren't really good either, because often we were guessing the wrong gene, or maybe we weren't genotyping enough markers in that gene. We didn't have sample sizes big enough to detect those small effects. And then, in about 2007, something called genome-wide association entered into the picture. So, a genome-wide association study is basically, it's a little bit like a genome-wide linkage analysis, but rather than just 400 genetic markers, now you're using like 1 million genetic markers across the genome. So you've got a much finer scale resolution than what you have with linkage analysis, and it's also an order of magnitude more powerful than linkage analysis when it comes to common disease genes. And so 3 things happened that enabled genome-wide association studies. The first was the publication of something called the HAP map. So the HAP map is like a big heat map. It's like looking at the correlation between genetic markers right across the genome. Why is this important? Well, it's important because instead of having to sequence 3.3 billion base pairs of DNA for everyone, if you know what the correlation structure is and the redundancy in it, all you have to do is look at 500,000 genetic markers, not 3.3 billion of them. And so the HAP map allowed us to survey genetic variation in an efficient way. Also, the advent of high-throughput genotyping. So this little SNP chip that you can see in that photo, that allows us to genotype one individual on 500,000 genetic markers. So now we had this technology that was available to do this, and it was also economically feasible as well. The epidemiologists had also provided us with massive cohorts of patients with blood samples that we could then genotype on these genotyping chips and then subsequently meta-analyze in these absolutely massive genetic studies. So this is one of the first ones. It's called the Wellcome Trust Case Control Consortium. It's essentially a big genome-wide association study of 13 different diseases, I'm going to say. So 7 Manhattan plots are shown in this figure here. So as Helen was saying, the y-axis is your statistical significance, your x-axis is the genomic position, and when you have a tower, like in Manhattan, that's telling you the gene under here probably has something to do with your disease. So this is just like kind of a little timeline of what happened with genome-wide association studies. So this was back in 2005. You can see chromosomes 1 to 22 in the XY chromosome. And whenever there's a genome-wide significant finding, there's going to be a little flag there. So technically, the first one is a very tiny study back in 2005 looking at age-related macular degeneration. Only had about 100 individuals. It was very sparse, but they did find something significant and replicable. But you can kind of see, as time's going on, a few more. First quarter of 2007. Then the Wellcome Trust Case Control Consortium was published. Loads more. You get the idea. And this is like back in 2019, and I've run out of space. Okay, so we're now in the situation where we have literally hundreds of thousands of genetic associations with common complex traits and diseases. And I feel like, you know, if you study the history of medicine, you're going to be hard-pressed to find another period that we've been doing science with such a concentrated flurry of discoveries, robust discoveries, across such a wide spectrum of diseases as with this age. Okay, who cares? The number one reason that GWAS is important to my mind is it tells us something important about the biology of disease. Because it's a hypothesis-free approach, it can tell us things about disease that we had no idea about. Here's some examples that I've put on this slide. I'm going to talk about our old friend ankylosing spondylitis. So this was a GWAS. It appeared in the first Wellcome Trust Case Control Consortium. I was very fortunate to be involved in it. Matt Brown, colleague of mine, was probably the main PI in these studies. You can see our friend HLA-B27 right in the middle. That goes way up to the sky, that particular hit. The one I want to direct your attention to is this circle here in a gene called interleukin-23R, or the receptor for interleukin-23. Why is this important? Well, it happens that IL-23R sits on the cell membrane of a particular sort of cell in the immune system called a Th17 lymphocyte. Th17 lymphocytes, they're found in the gut mucosa and other tissues, but a lot there. And they secrete the inflammatory cytokine interleukin-17. Okay? Before our study, immunologists didn't know this. So this is telling us something very, very important about the underlying biology of ankylosing spondylitis. But it doesn't stop there. Because when we made this discovery, the pharmaceutical companies had already developed a monoclonal antibody that was targeting interleukin-17. And they were trialing it for other autoimmune diseases with varying degrees of success. And so then this GWAS came along and said, hey, you should be trialing it in ankylosing spondylitis as well. which happened, and the controlled trials were positive, and then Novartis, the company that had the patent on this molecule, secukinumab, they got FDA approval to use it to treat ankylosing spondylitis in 2016. So this is kind of one of the flagship stories from the GWAS world of a finding that's allowing what's called a drug repositioning. So you had an existing pharmacotherapy, and on the basis of a GWAS, you trialed it for something else on the basis of that information. So there's this massive problem in the pharmaceutical industry called late-stage failure. And this is the idea that compounds that initially look really promising in animal models, phase 1 clinical trials, when they get to phase 2 and even 3 clinical trials, they either lack efficacy and don't work, or they have unwanted side effects. And so genome-wide association studies are one of the best ways of predicting whether your molecule is going to do something in terms of efficacy. You can also use it to look at side effects as well. So here's a number of other examples of successes that have either arisen from genetic studies or specifically genome-wide association studies. This list is only going to increase. It takes at least 10 years to, develop and test drugs. So this is not a quick process. Okay, so let me move on to something that Helen was talking about this morning, and that's this idea, can genetics tell us something about modifiable environmental causes of disease? And as you've seen, the answer to that question, I think, is yes. So this is a paper from 2003 that was led by George Davey-Smith and Shah Ibrahim, which kind of formalized this idea of Mendelian randomization, which has really taken off now that genome-wide association studies have found variants that we can use to proxy environmental exposures of interest. So Helen talked a little bit about this. I'll also quickly go through this slide. So if we were interested in, does smoking cause lung cancer? We can't do an RCT of that, right? And so we can't physically randomize individuals into a treatment group where we force them to smoke for 30 years, another group where we give them a placebo, get them to suck on a lollipop for 30 years, lock them in a room. And this randomization, that's really the reason that gives the RCT its robustness against confounding, because the randomization process ensures that our two groups are matched in terms of both known confounders, but also unknown confounding variables as well. And so when we measure the incidence of lung cancer between those two groups at follow-up, if the trial's big enough, we've done it competently, then we can pretty much say whether smoking causes lung cancer. Mendelian randomization, it's a very, very similar idea, but rather than physically randomizing individuals, we let Mendel's laws of segregation and independent assortment do the work for us. So in this particular example, we can take a genetic variant that we know is related to how much people smoke. So because of GWAS, we know heaps of these things. So there's one in a gene called the nicotinic receptor. So if you have this variant, you tend to smoke more cigarettes per day, you breathe in more deeply to get a bigger hit from the nicotine. We can use that variant and then use its random segregation and independent assortment to divide our sample in theories into a group who are predicted genetically to be heavier smokers and a group that are predicted to be lighter smokers. Because of this randomization, again, we expect that the known and the unknown confounders should be matched across the groups. And so if there's a difference in incidence or prevalence of lung cancer between the two groups, then that's some evidence that smoking causes lung cancer, and you don't have to do a randomized controlled trial for 30 years. Remember this guy? So this was Ronald Fisher from 1918 who had that seminal paper. Well, here he is again in 1952, a little bit older. He actually kind of realized this, okay? Although George and Shah formalized the idea of Mendelian randomization, Fisher's a smart guy. Like, he knew that genotypes were special. So if we have a read of the highlighted phrase, genetics is indeed in a peculiarly favored condition in that providence has shielded the geneticist from many of the difficulties of a reliably controlled comparison. The different genotypes possible from the same mating have been beautifully randomized by the meiotic process. A more perfect control of conditions is scarcely possible than that of different genotypes appearing in the same litter. Fisher was big on animal studies. Generally speaking, the geneticist, even if he foolishly wanted to, could not introduce systematic errors into the comparison of genotypes because for most of the relevant time, he has not yet recognized them. What a beautifully controlled experiment. So Fisher got it, and this is back in the 1950s, and you know, we're 70 years behind him. So here's some successful examples of Mendelian randomization. So things like LDL cholesterol, that's causal for coronary heart disease, but HDL cholesterol, probably not. C-reactive protein, an inflammatory marker that the pharmaceutical companies were interested in for a long time, actually, that's not causal for coronary heart disease. Don't waste billions of dollars developing an agent targeting CRP because Mendelian randomization is telling you it's not going to work. We play a little bit in this space. Helen's very nicely shown us some work this morning in the DOHAD field. One of my postdocs is interested in nutritional epidemiology, which is a notoriously difficult subject that's plagued by these issues of confounding. My go-to journal for everything in nutritional epidemiology is the Daily Mail. So drinking 2 or more cups of coffee each day may double your risk of heart disease, death, but only in people with very high blood pressure. A little bit later, actually, study pinpoints exact number of coffees per day that can cut the risk of sudden cardiac death. So actually, coffee's good for you. But apparently only if you drink it at a specific time of day. Who knew? So that's giving you an idea of some of the challenges. And so we thought about this question and how we might be able to design a Mendelian randomization study to investigate it. This is one of my postdocs, Daniel Huang. He's got an interest in what he calls chemosensory perception. I don't really know what that is, but it's got something to do with olfactory and taste receptors. And so what he's doing is he's using genetic variants in these olfactory and taste receptor genes to proxy whether you like foods or not, and also whether you eat more of them, okay? The advantages here being we understand the biology a little bit more of why these genetic variants are related to how much of a particular food we consume, and also importantly, when folks have tried to do Mendelian randomization studies in the past in this space, they've taken GWAS of, like in the UK Biobank, how much of this food do you eat? How much of that food do you eat? We think that's a problem because A, you don't usually know what the genetic variant is doing, but also importantly, the results of these GWAS often represent reverse causality. So imagine, you know, you like your Big Macs, okay? You go to your doctor, the doctor says, you got high cholesterol, cut down on the Big Macs. What's going to happen? Your variants that predispose you for coronary heart disease are suddenly going to be related to how many Big Macs you eat, right? So we think that this is a more robust way of looking at these questions in nutritional epidemiology. So the panel in the center lists all the different receptors, either olfactory or taste, and then the specific components of the diet that they're associated with. So some of them, it's just one. Most of them, it's more than one, or some even lots of. And so the idea is to use those variants in various flavors of Mendelian randomization analysis to investigate diet-related causes of disease. Here's one finding that I've cherry-picked. So there's a variant that's really strongly related to how many onions you consume, whether you like onions or not. It's not related to a lot of other things. And when you look across a range of different health outcomes, that same variant that's related to how many onions you consume is related to decreased risk of systolic blood pressure, of hypertension, and decreased systolic blood pressure and diastolic blood pressure. So onions are good for you? Maybe. This is going to be lost on everyone who's not Australian and New Zealand, isn't it? This is our former Prime Minister who once famously chewed into a raw onion live on television. But he's probably got very low blood pressure, so... Okay, so that's kind of a bit of a whirlwind tour of the last 150 years of genetics. I don't have much time now to say where we're actually going. Here's a few things. So, as you saw, there's tens, hundreds of thousands of these robust genetic associations. It's now going to come back to the molecular biologists to work out what these associations mean. How are they exerting their effects on risk of disease? Once again, amazing technologies are being developed all the time. So single cell sequencing, this is the idea of we can quantify the expression of genes at the individual cell level. So we can see what genes are likely important and what cell types as well. It's phenomenal. You would have heard of CRISPR-Cas9 as well. So this is the idea that you can take a DNA sequence and you can perturb that sequence experimentally. You can make a change. So again, that's very exciting in terms of working out mechanisms, but also there's starting to be therapies as well in the clinic involving CRISPR-Cas9. Stratified medicine, so I haven't called it personalized medicine because I don't believe in personalized medicine for most things. Some things I do, most things I don't. Stratified medicine is the idea of stratifying people by their risk. So for some diseases, we're explaining so much of the variance in their underlying risk that we can do a pretty good job of putting people into high and low risk groups for screening. Got no problem with that. I think that'll be good. In terms of, saying you're going to get disease or you're not, no. Like that's never going to happen because of that gloomy prospect for most things. AI and machine learning is also really exciting. Come next week. There's a really good course on AI and machine learning. I know nothing about it, but people who do are teaching the course. And longitudinal studies. So this is, integration of wearables with really high dimensional phenotype data. I'm sure there's loads of other things as well. So, at the beginning of the talk, I asked, what did the geneticists ever do for us? This is a Monty Python reference for folks who are under 50. Hopefully, I've convinced you that geneticists have allowed us to say something about the nature versus nurture controversy. GWAS has taught us about the underlying biology of disease, and in some cases identified targets that have resulted in either new drugs or repositioning of existing pharmacotherapies, and Mendelian randomization studies have helped establish or refute causal relationships between risk factors and disease. I just want to finish by acknowledging funders, different cohorts, and I just want to call out 3 people who've been very important in my career as mentors. So Matt Brown, who led most of the work in ankylosing spondylitis. Lon Carden, who was my postdoc advisor at the Wellcome Trust, who got me involved in the International HapMap project, the Wellcome Trust Case Control Consortium, and a face many of you will know, George Davey-Smith, who, after a long process, taught me how Mendelian randomization worked. It took like 6 months or something, but he got there eventually. Daniel Huang from my group, who's doing the nutritional epidemiological work, all the members of Evan's group, and last but not least, Odin for making sure that the conference calls don't run over time in the evening. Thank you very much.